Hello, my excellent biologists. We are going to finish our discussion on chapter 27. We've already talked about the early Earth and its formation. We've talked about the importance of the mantle and those convection currents making the plates move around and the dynamics of that. And now we're going to talk about a hypothesis about how life could have formed on this early Earth in those conditions. Remember, we don't know. These are the best ideas based on what we know through science and scientific evidence. This is not based on any type of supernatural action to create life here on this planet. So let me go ahead and present, here on our story of life. All right. So the first thing we want to talk about is what would the atmosphere have been on that early Earth? Remember, the Earth is 4.6 billion years old, and you didn't have your first living cell until about 1 billion years later. Okay, so what were the conditions on this early Earth, and how would have that contributed to the formation of life? So the atmosphere that you would have seen on this early Earth would have no free oxygen, meaning if I could time travel you back there, Umbrella Academy, you would not, cultural reference, sorry, you would not have been able to survive, right? Because there was no free oxygen. Now, the gases that were probably in the atmosphere, remember all the plates are moving around and you have subduction zones, you would have these volcanoes, and the outgassing of those volcanoes would have been the atmosphere for that early Earth. So so here's a picture, of course, because somebody was there, right? So here you can see, like, there would be water, right? And then the gases, so you had rain, and then the gases that came out of those volcanoes would not have had any oxygen in it. This would be considered, and by the way, I forgot an S right here in similar. Let me get my pointer so you can see. There should be an S right here. This would have been considered a reducing environment, and a reducing environment is very conducive to building, for molecules joining together. Today, currently, we have an oxidizing environment. Things tend to break down in our environment because oxygen is so electronegative and pulls away those electrons. So now we have an oxidizing environment, but early Earth was a reducing environment. There wasn't oxygen in our atmosphere until about 2.3 billion years ago due to the oxygen revolution, and this was when small, probably like cyanobacteria, gained the ability to split water when they did photosynthesis, releasing oxygen gas. And we'll talk more about that in just a little bit. So what would the gases have been? Would have been something like what you're seeing right here. Notice, again, I'm pointing out, there is oxygen in something like carbon monoxide, but not free oxygen like O2, what you're breathing today. There would have been oxygen in the water, H2O, but not free oxygen like O2, like you're breathing today. So on your notes, on stage 1, the evolution of small organic monomers, and I'll explain that in just a minute, you want to know the early Earth, the beginning of the Earth was about 4.6 billion years ago. So put that in your notes, 4.6 billion years ago. It was a reducing environment, which meant very little free oxygen, very little free oxygen. And this was, letter C, good for building molecules. And then you probably want to add on there, whereas now it is an oxidizing environment. Things tend to break down. Okay. The substrate, these are gases similar to what would have been released from a volcano. What would have been released from a volcano. Now, none of these molecules that you're seeing right here are considered organic molecules. Carbon, when you have carbon in something that makes 4 bonds, then that is what would be considered an organic molecule, not like organic vegetables at the store. Organic meaning built out of carbon. This carbon monoxide, carbon dioxide, these are exceptions to organic molecules because the carbon is so simple here. So let me show you what I mean. Hopefully you can see, like, these are stairs, like here's stair 1, stair 2, stair 3, stair 4. We're going to be talking about all of this and parts of cells, etc., when we start progressing through here in Unit 2. But what I want you to see right now is inorganic molecules, that's what was on the early Earth originally, right? That is what came out of those volcanoes, those gases. And from that, how would we go from inorganic molecules to organic molecules? The organic molecule you're seeing right here, this is called an amino acid, and amino acids build proteins. So that would be a macromolecule, would be a protein. And then we use these macromolecules to build cells. So things like the four important organic molecules are proteins, carbohydrates, lipids, nucleic acid. Nucleic acids are things like DNA and RNA. So if I were to kind of annotate this diagram a bit more, okay, what I want you to see is early Earth was stair one. Early Earth, you just had these outgassing from volcanoes, and this is what you had to work with. So how did you go from here to the second step where you had simple sugars and amino acids and fatty acids? Well, the only thing you would have to build those is the energy in the early Earth. Energy, things like UV radiation and lightning, radiation from the Earth's core, those types of things would provide the energy to go from these inorganic to organic molecules. Once you have these simple organic molecules, then you would need, with the same energy I just mentioned, build these macromolecules, these polymers like proteins and carbs and lipids and nucleic acids. Once you have these important macromolecules, then you could start to begin to build cells. All right, so let's go to next slide here. Okay, so this prebiotic soup, let me make myself smaller. Okay, so as the early Earth cooled, you would start to collect these organic molecules in the soup, the ocean water. How could that be? Well, like I'm mentioning right here, energy from lightning and UV radiation and the Earth's core could have begun to build the simple organic molecules and later the large macromolecules. So if you look right here, in order to build these simple amino acids and simple sugars, you would have radiation, lightning, UV light, and then this energy could have formed these simple sugars. So let's put in our notes, energy, number 3, UV light, molten magma, lightning, radiation. And then you would think, okay, yeah, I don't believe this. I don't think this could happen. Well, let's look at an experiment that was done. This was an experiment suggested by a man named Operin, but was completed years later by a man named Miller. And what he did is he tried to recreate Earth's early environment. So here would be like your ocean, right? And you would have energy here. They have an electrode right here. So this would be like lightning, right? Or energy right here from the Earth's core, the sun, UV radiation. And he put the gases in this apparatus that were similar to gases like that would come out of a volcano. And he circulated these gases, and literally, okay, within less than 2 weeks' time, when he checked to see what was in this apparatus, he did build all those simple organic molecules like amino acids, fatty acids, glycerol, and nucleotides. So remember, the Earth is 1 billion years old, and it took about 1 billion years before you have any fossilized life form. And yet within less than 2 weeks, he was able to generate that, go from step 1 to step 2, where you get just organic molecules. So on your note for your experiment, this was proposed by Operin. It was performed by Miller in 1953. And he circulated proposed gases in an apparatus modeling the conditions of early Earth. After just 1 week, so just about less than 2 weeks, after 1 week, he had amino acids, simple sugars, and other small organic molecules. So after 1 week, he had amino acids, simple sugars, and other small organic molecules. So then if you're looking at that first hypothesis, step 1, evolution of small organic molecules, what's being said is within less than 2 weeks' time, just a little over a week, you could go from just the inorganic gases to the small organic molecules. So that's one idea. There are a couple other ideas here as well. And so number 5 on your notes also proposed that organic molecules had an extraterrestrial origin, not like ET coming and landing, but like comets and meteorites. So comets and meteorites, maybe they seeded when they came from other areas outside of our Earth, would have seeded the Earth with organic molecules. So that's another hypothesis. All right, stage 2, the evolution of polymers. Polymers would be things like complex carbohydrates and lipids and nucleic acids and fats. So the evolution of the organic polymers from monomers, one of those ideas is worried about like the source of nitrogen, that in order to make those polymers that have nitrogen in them, is where would that have come from? And what is hypothesized that minerals that form at deep sea vents, like this one, could catalyze the formation of things like ammonia. Remember when we talked about nitrogen, there wasn't any life to do nitrogen fixation. So what would be your source of nitrogen? So you could do nitrification, right? So the formation of ammonia and other organic molecules would have been the heat of the earth. And then what is right here within the minerals of these deep sea vents that you could catalyze then N2 into ammonia. Okay, and we talked about that with the nitrogen cycle and how we need nitrogen fixing bacteria to give us a large enough source of ammonia that other bacteria can do nitrification, right, to form nitrate for plants. So if you have no life, no nitrogen fixing bacteria, where is that going to come from? Catalysts like iron, nickel sulfides and rocks at hydrothermal vents could form that ammonia. So on evolution of organic polymers, energy at hydrothermal vents forming ammonia. All right. Also on your notes, why don't you add on where it says stage 2, evolution of organic polymers, let's list what those polymers are. Complex carbs, proteins, lipids, and nucleic acids. Complex carbs, proteins, lipids, and nucleic acids. Those are the polymers we're trying to form. All right, and then where could all of this have? So now we have these organic molecules. How do you put those amino acids together to build a protein, or sugars to put together a carbohydrate? And what is hypothesized is the same energy that was used before, right? UV radiation, lightning, right? The Earth's core generating heat, that could have been the catalyst, right? That could have been what formed those bonds. But also the surfaces of clay rocks can also hold energy and re-release it, and that could have also acted as a catalyst to form those large macromolecules. So number 2, proteins first when heated in shallow pools. So you could have formed proteins when heated in shallow pools and on the clay surfaces. So clay surfaces acting as a catalyst. Catalyst, C-A-T-A-L-Y-S-T. All right, so let's take a look right here, okay? So I want to see where we are. So inorganic molecule, outgassing of volcanoes. How do we get to organic molecules to take that first step? UV, lightning, right? Molten magma, radiation from the Earth's core could have formed those organic molecules. Or it could have been seeded from comets or asteroids. An experiment was done where you go from step 1 to step 2 by Miller, and in less than 2 weeks, they got organic molecules. Going from step 2 to step 3, going into the polymers, again, same energy as before, molten magma, radiation, right? Also, clay surfaces do act as catalysts to take those simple organic molecules and build more complex polymers. And where you see this already is there are things called microspheres, and this is when hot proteins are cooled rapidly. They will form these sphere shapes, these protein spheres. And then there's something called Operan's quacervate droplets, which is when fats form spheres. And this could have been the beginning of where you're forming now a cell, an early cell, because an early cell has a phospholipid bilayer with cells have a phospholipid bilayer with proteins embedded in them. And so what would happen, how could you have formed that cell membrane? Well, already you can generate these quite rapidly is just this fatty layer from lipids in water, and hot proteins cooled quickly will also form layers. So this could have been the beginnings of stage 3, the evolution of protocells. So back to where we are, inorganic to organic, organic to macromolecules, polymers. Now we're trying to build that first cell, and the simplest of cells would be a prokaryote. And we'll talk more about prokaryotic and eukaryotic cells later. So the thing that has to happen is you have to have that outer membrane first. So evolution of protocells having at least an outer membrane, having at least an outer membrane. Okay, so now, to get to a true, from this protocell, right, to get to a self-replicating true cell, there's several steps that need to happen, okay? So the first cell was probably about, that first cell was probably about 3.6 billion years old, okay, when you had your first true self-replicating cell. That first cell was more than likely a heterotroph. Remember, we talked about ecosystems. Heterotrophs consume nutrients. It's easier to consume, right, than to do the step where you take inorganic CO2 and fix it into sugars, right? That's another whole step. So the first cell was probably heterotrophic, where it was just consuming nutrients. Then autotrophs evolved, and then a type of autotroph that actually split water and released oxygen, creating the oxygen revolution. That was about 2.3 billion years ago. And then these simple prokaryotic cells evolving into eukaryotic cells, and I'll talk to you about the endosymbiotic hypothesis later about how that could have happened, and then going from single eukaryotic cells to multicellular organisms, and these multicellular organisms then moving on to land. And we'll talk more in depth about that process later, but what I want you to see on stage 4 is evolution of true living cells having a self-replication system, and more than likely this was RNA based, and we'll be talking about that a little bit later, most likely using RNA initially because it can act as a substrate, meaning it could hold the genetic information, right? And it can act as an enzyme. So what I want you to add to stage 4, evolution of true living cells, is this right here. Your first cell is about 3.6, 3.5 billion years ago, then a heterotroph, right? It's consuming nutrients, then an autotroph, then you have the oxygen revolution about 2.3 billion years ago, and then you would have your first eukaryotic cell and then a multicellular organism. So you might need to hit pause right now so you can add that to stage 4, all right? But I'm gonna keep going, so hit pause if you're still writing. Okay, so like I said, the first fossil about 3.6 billion years ago, a prokaryote, heterotrophs, and then later autotrophs, and then later eukaryotic cells and multicellular organisms. Okay, and there's a picture in your book showing this about the chemical evolution versus the biological evolution. So you can see early Earth, inorganic molecules, small organic molecules, the beginnings of macromolecules in a cell membrane, an early cell, and then a replicating cell. And we'll be learning all about cellular respiration and photosynthesis. And these, my friends, are the steps to life.